Hello, welcome to our project on predicting protein domain superfamilies. My name is Laura Alvarez.
And my name is Carlos Guerra, and let's begin by going over the background and motivation behind our project. So, what is a domain, what is a protein superfamily, and why does it matter?
To start, a protein chain itself is a sequence of amino acids where an amino acid is a basic organic molecule, and these amino acids fold into one another to create local structure, and these local structures then fold into one another to become a global structure. And it's this global structure that we'll see later on that is of interest to us. Now, a protein chain can be further decomposed into domains, where a domain is a segment of the protein that can evolve independent of the rest of the protein chain and is also independently structurally sound. These domains can then be grouped together into sets called superfamilies, and these superfamilies are notable insofar that their members are similar to one another in global structure. Now, it's important to note that a protein shape dictates its function within your body. For instance, this protein is acting as an enzyme due to the shape of its active site. Now, proteins can have other functions, for instance, carrying oxygen in your blood or carrying other nutrients due to its shape. Now, knowing the functions of these proteins is essential into understanding diseases as well. This observation that shape implies function fuels our main motivation for this project, in that if we are able to predict the superfamily of a protein's domain, then we can further deduce the protein's overall function, which in turn allows us to make new discoveries in terms of what causes disease or how to combat disease. So what work has been done thus far and has influenced our approach? Well, to begin with, we need to go over what structural protein data is. When structural protein data is just a sequence of XYZ coordinates for each atom in the protein. And these have the following properties: that they're spatial, temporal, and their lengths can differ for different proteins. And if we could remove all of the excess atoms aside from the alpha carbon backbone of a protein, we can dramatically reduce the size of the data while still preserving the structural information that we are interested in. This leads us to the frag bag representation, which is essentially a bag of words representation of a protein that was proposed in 2002 by R. Kolodny. The idea is that we can construct an F-size library of protein segments or fragments that are common within nature, and where each fragment has n number of alpha carbons. We could then use the following algorithm in order to vectorize any given protein P as a vector of length f of the frequencies of any given fragment within our database. And we do this by iterating through every n consecutive alpha carbon block within P. This has the added benefit of preserving some local structural information, similar to how in text mining n-grams can preserve local syntactical information. Now, this process utilizes this similarity metric called LMRSD, where RMSD is the average distance between alpha carbons between two proteins, and the LMRSD is the RMSD of the superimposition such that the RMSD is minimized. This project is based upon a paper that also did superfamily prediction utilizing frag bags, albeit with fragments of longer lengths and a larger library. Now on to data retrieval and pre-processing. So we begin the data retrieval by issuing a PDB query, which is the protein databank, and from which we get the atom coordinates of a protein, and we can then dissect that into just its alpha carbon backbone. And then we use scope E, which is the structural classification of proteins extended, to get the domains of all the alpha carbons within that protein.
The dataset is made of nearly 12,000 domains and represented by 250 features. Our machine learning problem is a multi-class problem with 7 classes, one for each superfamily. This graph shows the distribution of the families along the data. As we can see, we are dealing with an imbalanced problem where the 3 main classes made up the 70% of the entire dataset. The graph on the left shows the accumulated variance over the principal components. To preserve 90% of the variance, we reduce our features to 130. To perform this dimensionality reduction, we experimented with the following techniques: PCA, linear kernel PCA, SVD, and autoencoders. The best results were given by PCA and SVD, but we will discuss more on this later.
In our experimental setup, we utilize the smote sampling technique in order to oversample underrepresented superfamilies and undersample overrepresented superfamilies. We also utilize repeated stratified threefold cross-validation. We utilize the stratified version in order to preserve the proportions of each of the classes in each sample. And we utilize 5 repetitions, meaning 15 trials per classifier. We then use the weighted F1 score, again because of the imbalance in classes, to evaluate each of the classifiers. We then use accuracy, the false positive rate, and the true positive rate on each of the superfamilies for the best classifier.
Now, with this experimental setup, we must determine which classifier performs the best.
Our neural network design contains 2 hidden layers, each using ReLU as their activation function and as an output function using softmax. And this uses categorical cross entropy as its loss functions, and each of the hyperparameters were found through a grid search.
Decision trees. To fit the parameters of our classifier, we use grid search. From the grid search, we got that the classifier performs better when the max step of the tree is the number of features of the data. To measure the quality of a split, we compute the Gini index. The next classifier we used was KNN, with 5 neighbors. The Euclidean distance as a metric to measure distance between points, and a distance weight voting. The last classifier we would like to talk about is singular vector machines. We also performed grid search on this one. To our surprise, the linear kernel performed the best between all the different kernels. The results will be discussed later on. Now, we would like to analyze the performance of all the classifiers. The columns of this table represents the dimensionality reduction technique that we applied, and the rows are the classifiers. The evaluation is done with the F1 score and the runtime. As we expected, the best performance was obtained without any dimensionality reduction using neural networks. On the other hand, by applying a dimensionality reduction technique, we can reduce the runtime at 64% and still obtain an F1 score of 0.98. With F1 score, we can deal with the unbalanced nature of the data. Also, the performance of the neural networks is good on the non-reduced data because it can perform its own feature selection. As we can see, our neural network outperformed the SVMs, especially in that the represented classes. This is surprising due to we are using a smaller fragments library. We expected to lose some information. The reason of the results could be our sample technique using a SMOD that can represent better these classes. Structural findings. What did our project reveal about the structure of the data? After our analysis, we can conclude that the data is generally linearly separable data. In all of our tests, the kernel linear outperformed the other kernels. Also, the good performance of naive classifier as KNN suggests that ours is well-separated data. Ergo, FragBack is a good representation of our protein structure, and somewhat we have well-clustered data when we run k-means using Euclidean metric. This is a TSNE visualization of the data, where we can see the data is generally clustered. And again, that could be one of the reasons why naive approaches as KNN work so well. Our main conclusions are that our classification technique performed better than in 3, and also it has been shown that FragBack structural representations preserves enough information for data to be well separated.
Hope you enjoyed our presentation.
Thank you.